Good morning. I am Professor S.K. Khare from IIT Delhi under the EPG Pathshala of UGC in the subject biochemistry. Today we are going to discuss about the messenger RNA, their structure and function. The structure of mRNA or messenger RNA is not very well defined. In essence, this is complementary to the DNA strand from which this is transcribed. So, the secondary structure sometime it acquires a hairpin kind of a structure. However, there are three specific features in the mRNA which makes them very distinct. One is a 7-methyl guanosine cap at the 5' end and then a poly A tail at the 3' end of mRNA. This feature is very distinguished feature of mRNA. mRNA has a role in initiating and then providing the codes for translation. These codes are in form of codons, the message coming from DNA. The binding or initiation of transcription is decided by mRNA based on a sequence called Sine-Dalgarno sequence, which is upstream in the mRNA. These SD sequence binds to the complementary sequences in 16S rRNA subunit sequences of ribosome. The objective of this lesson is to understand what is mRNA or messenger RNA. We will also learn in this chapter the difference between prokaryotic and eukaryotic mRNA and protein synthesis. We will also look into the structure of mRNA and role of 7' methyl guanosine cap, 5' UTR, 3' UTR and poly A tail in protein synthesis. What is mRNA? mRNA or messenger RNA is a working copy of DNA. This acts as messenger between DNA and protein. It transfers information from DNA to synthesize the protein with the help of rRNA and tRNA. The structure of mRNA is not very well defined. However, sometimes it acquires a hairpin like structure. However, you will see certain structural features which are essentially a part of mRNA. One is that you will see in this picture a 7-methyl guanosine cap on the 5' end, the green one. The structure of this 7-methyl guanosine is given here. It is a methylated guanosine, and then you will also see a poly A tail. Poly A tail means number of adenosine residues at the 3' end of the mRNA. So, you can here see typically the structure of mRNA. What is shown in this picture is the difference between prokaryotic and eukaryotic mRNA. In both the cases, you will find 7' methyl guanosine cap, a 5' UTR, cistron 1, 2, 3 and then 3' UTR in case of prokaryotic mRNA and then a poly A tail. But in case of eukaryotic mRNA, what is missing is 2 and 3 cistrons. Let us now discuss the role of mRNA in protein initiation in case of prokaryotes. In the upstream of prokaryotic mRNA, a purine-rich sequence called Sine-Dalgarno sequence or SD sequence is present, which is ACC UCC UUA. This sequence pairs with complementary sequence present in 16S rRNA of a small ribosomal subunit. The sequence consists of 3 to 9 adjoining bases in mRNA that forms standard base pairing with some or all the bases at the 3' end of 16S rRNA. In the picture you can see this kind of pairing between Sine-Dalgarno sequence and the 16S rDNA. The spacing between SD sequence and the initiation codon varies with average of about 7 nucleotide, and this distance determines the efficiency of translation. We will next discuss the working of polycistronic mRNA in prokaryotes. In polycistronic mRNA, translation initiation can occur at multiple sites and translation may be affected by the neighboring cistrons. However, translation initiation first takes place with that cistron which has a stronger ribosomal binding affinity. This phenomena is regulated by Sine-Dalgarno sequence or AUG bonding as discussed previously and a part of preceding coding sequences. The base pairing is disrupted when ribosome advanced through the upcoming cistern as a result of downstream cisterns get activated. Thus, elongating ribosome disrupts secondary structure which is inhibitory to upcoming ribosome. Translation coupling is the important mechanism that coordinates the synthesis of ribosomal protein. In some cases, some ribosomal subunits are used for upcoming cistrons. This mechanism is called reinitiation. There is a vast difference between reinitiation in prokaryotes and eukaryotes. In prokaryotes, there is no limitation on the size of upstream cistrons, and also the end of first cistron often overlaps with the start of the second. The similar arrangement is not possible in eukaryotes. Few studies on E. coli showed that following termination, the loosened ribosome may move forward or backward to locate reinitiation site. The role of mRNA in protein initiation in case of eukaryotes is different from prokaryotes. Smaller ribosomal subunit having methionine tRNA, elongation factor ELF2 and GTP first enters to the 5' end rather than at AUG codon. They then migrate through 5' UTR till they reach to the AUG codon to take a pause, where 16S ribosomal subunit joins with 14S and fixes the selection of the start of codon. In eukaryotes, flanking sequences determine the efficiency with which the first AUG codon is recognized and act as a small pause during scanning phase of initiation. Vertebrate mRNAs traditionally have consensus sequence flanking AUG is GCC, RCC, AUG, G5', 3'. Within this consensus sequence, the purine are positioned as 3' and T from first A of AUG is highly conserved. Usually at position A3 is found in most of the cases. AUG codon can be a weak or strong start based on the presence of nucleotide at downstream 3 or upstream 5 position. However, these consensus sequences are recognized and how they function is still not known, but it was predicted that it works by scanning mechanism. Translation of most of the mRNA depends upon 5' M7G cap. The leaky skinning occurs due to the absence of good context around the first AUG codon. Almost 2 dozen mRNA were found to have 5' proximal AUG codon present in suboptimal context. Thus, ribosomes initiate both at first and second AUG codons, producing 2 variable proteins from one mRNA. Leaky scanning may also occur when initiation starts at a non-AUG codon such as ACG, CUG or GUG. However, in higher eukaryotes, pairing between non-AUG codon and methionine tRNAi is weak. Only alternative codon that have good base pairing are usable in mammals. This picture shows the mechanism of leaky skinning, and you can see the dashed line shows the pathway of 14S subunit. The usual restriction of initiation to the first AUG codon can be avoided by reinitiation, the line 2, or context dependent leaky scanning, line 3. So, to summarize, what we have learned is that mRNA functions for the translation, and mRNA is generated from DNA from the process of transcription. It is the copy of DNA strand from which is transcribed, thus carry the coded information for translation. However, there is a difference between prokaryotic mRNA and eukaryotic mRNA. So, in this lecture, so far we understood that messenger RNA are very important. They are transcribed in the nucleus, then they are transported to the cytoplasm, reaches to the ribosome where translation takes place. They are carrying information in form of codons. Their structures are unique in having a 5' cap and 3' tail, and then they have a specific recognition to bind to the ribosomal subunits. This is what we have understood in this lecture so far. The prokaryote and eukaryotic mRNAs are different.